In this module, we discuss electron transport, the stage in aerobic organisms that's coupled to making most of the ATP in respiration. In an earlier presentation, we saw that after glycolysis and 2 turns of the Krebs cycle in aerobic organisms, the free energy that was once in glucose is in 10 NADHs, 2 FADH2s, and 4 ATPs. So in this module, we'll see how the free energy in the reduced electron carriers is released during electron transport and captured in a gradient of protons. In a later module, we'll see how relief of this proton gradient is coupled to ATP synthesis by oxidative phosphorylation. First, a reminder about redox reactions. In this generic reaction, electrons are being transferred as part of hydrogen molecules, a characteristic of many biochemical redox reactions. The reduced and oxidized forms of the same molecule are called a redox pair. From the equilibrium arrows, you can see that this reaction is exergonic, so it would have a negative standard free energy change, a negative delta G0, releasing free energy. How about this real reaction? Can you identify the redox pairs here? They are pyruvate and lactate, and NAD+ and NADH. Pyruvate and lactate are simply the oxidized and reduced forms of the same molecule, as are NAD+ and NADH. The transfer of electrons between molecules in redox reactions is a lot like the flow of electrons in an electric circuit. Take this simple battery, for instance. By convention, the plus button, or the plus end, is the anode, and the flat end of the battery is the cathode. When the circuit shown here is closed, electrons flow from the cathode to the anode. This is because the anode contains chemicals with a higher potential to be reduced than the chemicals at the cathodal end of the battery. The chemical reaction in the battery is spontaneous, releasing free energy. The electron transport system, then, is a series of molecules in the crystal membrane of the mitochondrion that can accept and then donate electrons. The electrons flow from NADH and FADH2 down this series of redox molecules because each subsequent component has a higher potential to be reduced than the one before it. Hence, the whole of electron transport releases free energy when the reduced electron carriers produced in glycolysis and the Krebs cycle are oxidized. This slide illustrates the redox components of the electron transport system, or ETS for short. NADH from the Krebs cycle is oxidized by a dehydrogenase enzyme containing FMN, that's flavin mononucleotide, another B vitamin-derived electron carrier. From there, its pair of electrons is passed as a hydride ion from component to component down the electron transport system until it joins with protons to reduce molecular oxygen to make water at the end of the chain. Likewise, FADH2 from the Krebs cycle is oxidized by the electron transport system, though its electron pair is accepted further down the chain by coenzyme Q, which is also called ubiquinone. Finally, NADH, formed in the cytoplasm during glycolysis, is also oxidized in the electron transport system. Since NADH can't cross the mitochondrial membranes to get into the matrix, chemical shuttles that can cross these membranes accept electrons from cytoplasmic NADH and deliver them to components of the electron transport system. So the entire electron transport system is an energetically favorable pathway, as shown here. As I noted, electrons are passed to components with higher and higher potential to be reduced, shown on the left. Well, the flow of electrons can be measured. Current is a measure of how many electrons are flowing, and voltage is a measure of the potential electrical energy of the electron flow. We can measure reduction potential. When we do this under standard conditions, the standard conditions that we would use to determine equilibrium constants and standard free energy changes, we will measure the standard reduction potential, or SRP, of a reaction. And this has been done for many of the reactions, if not all of them, in the electron transport system. And the graph in this slide plots the free energy and standard reduction potential of the electron system components, highlighting the fact that free energy and the standard reduction potential, or SRP, are directly proportional, and showing that there are 3 redox reactions with large drops in free energy. Take a look at those. In theory, the 3 big negative standard free energy changes that were greater than 7.3 kilocalories per mole are enough to fuel the synthesis of 3 ATPs by oxygen-consuming processes that came to be known as oxidative phosphorylation, or OP for short. Indeed, the stoichiometries of ATP synthesis coupled to NADH or FADH2 oxidation were worked out. NADH oxidation seemed coupled to the synthesis of 3 ATPs, while FADH2 oxidation fueled only 2 ATP syntheses. Based on the amount of these electron carriers produced in the electron transport system, one could predict that the free energy from glycolysis and the Krebs cycle could fuel the synthesis of 34 ATPs. You can do the math for yourselves using former balance sheets that I showed you. So now the question is, how is the exergonic electron transport system pathway actually linked to ATP synthesis, to oxidative phosphorylation? The first clues came from the effects of a respiratory poison called dinitrophenol, or DNP. Mitochondrial isolates could oxidize NADH or FADH2, consume oxygen, and make ATP, as suggested here. Mitochondria treated with dinitrophenol, while still able to oxidize the reduced electron carriers and consume oxygen, would not make ATP. Investigators concluded that the DNP uncoupled electron transport from ATP synthesis. In other words, the two processes were coupled but were actually independent processes. In what was a curious observation, DNP-treated mitochondria were able to hydrolyze added ATP. Curious because normal untreated mitochondrial isolates never exhibited an ATPase activity, an activity that would hydrolyze ATP. If a chemical, dinitrophenol, could stop ATP synthesis but leave the electron transport system intact, then it should be possible to physically separate the electron transport system from oxidative phosphorylation reactions. Ephraim Racker was experimenting with ways to dissect mitochondria and analyze the functions of the parts. This slide is a representation of the crystal membrane from electron micrographs, which suggested that the inner surface of the crystal membrane was coated with particles. The separation of the parts took Racker a long time to solve, but eventually his mitochondrial dissection was successful. So here's the dissection. Purified mitochondria were subjected to high-frequency sound waves, ultrasound. In the electron microscope, Racker and company saw fragments of membrane and membrane vesicles coated with stalked particles, which, when provided oxygen, ADP, inorganic phosphate or PI, and a reduced electron carrier, for example NADH, these broken mitochondrial bits were able to oxidize NADH, consumed oxygen in the process, and made ATP, just like intact mitochondria. Also, just like intact mitochondria, if dinitrophenol were added to the busted-up mitochondria, they would continue electron transport in the presence of DNP, but could not make ATP. Again, far from it, the vesicles would hydrolyze ATP. The stalked particles were then isolated by centrifugation and further dissected. An electron micrograph shown at the right shows the particles after this isolation. The supernatant from the centrifugation, containing mostly loose membrane bits, could not oxidize NADH or make ATP or do any of the things that mitochondria can do or that the original preparation can do. But the isolated stalked particles had all of the properties of intact mitochondria. They could conduct electron transport and oxidative phosphorylation, and upon adding dinitrophenol, these stalked particle isolates would do electron transport, but of course would make no ATP. And if you added ATP to dinitrophenol-treated particles, the ATP would be hydrolyzed. From his experiments, Racker concluded that the stalked particle was a crystal membrane fraction that had closed up inside out after isolation, and that the particles that coated the crystal membranes, these lollipops, were the same as seen in the electron micrograph in the previous slide. Because it had all of the respiratory activities of intact mitochondria, these lollipop-studded vesicles were indeed a model for studying mitochondrial respiration. Finally, Racker's lab was able to dislodge these lollipop particles from the membranes by shaking them in a high salt concentration solution. The remaining smooth vesicles moved to the bottom of a centrifuge during a spin, here it comes, while the stalked particles remained suspended. A single isolated stalked particle can be seen in the electron micrograph on the right in this slide. Fraction 2, F2, the smooth vesicle in the pellet, would conduct electron transport, but could not make ATP. Fraction 1, the isolated stalked particle, would not do electron transport, and of course would not do ATP synthesis. But if ATP were added to this fraction 1, the ATP would be hydrolyzed, leading Racker and company to call this fraction 1 an ATPase. So electron transport can be physically uncoupled from what is likely to be oxidative phosphorylation. Racker concluded that it was the F2 vesicle fraction that contained the electron transport bits, and then correctly surmised that the F1 ATPase lollipop particle was really an ATP synthase, but of course only when coupled to the electron transport system. Over time, the F1 ATPase particle became known as the F1 ATP synthase. And here's the conclusion. F2 has the ET parts, but no ATP synthase, and the F1 is really an ATP synthase. To make sure that the activities, or lack thereof, that Racker saw in the fractionated mitochondria were real, and not just some artifact of the mitochondrial isolation process, he tried to put the parts back together. He succeeded in reconstituting what sure looked like stalked vesicles in the microscope, in the electron microscope, complete with the activities shown here, including a recoupling of the electron transport system and oxidative phosphorylation, that is, the ability to make ATP while oxidizing NADH or FADH2, thereby regaining that ATP synthase activity. And, of course, the reconstituted vesicle behaved just like the original studded vesicle. In the presence of DNP and ATP, the ATP would be hydrolyzed. In the presence of DNP and NADH and oxygen, there would be electron transport, but even if ADP and inorganic phosphate were also added, no ATP would be made. That is, there would be no oxidative phosphorylation. These reconstitution experiments confirm that the components were not altered or inactivated by this mitochondrial dissection. I just want to point out that the idea of pulling apart organelles and subcellular particles and then putting them back together or reconstituting them is a recurring theme in cell and molecular biology studies. And so this concludes our module on the electron transport system.